This is the Anthrobiology Podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hello, everyone. Welcome to another episode of the Anthrobiology Podcast. Joining me via Zoom from Germany is Dr. Selina Carlhoff. How are you?
I'm very good, thank you. I am actually not a doctor yet. I'm just a PhD student.
That is true, but I'm so excited, and I know you're going to do it. And the reason I'm talking to you is actually because of a huge accomplishment that you had. You had your first paper in Nature.
Yeah, that's very exciting that my first project related to my PhD came out in Nature in August, actually, which seems a while ago now, but also still hasn't quite sunk in at the same time.
For sure. I don't know. It's such a huge accomplishment. I'm like, I don't even know you, and I'm so proud of you.
Yeah, I'm also very excited. It was weird to explain to my parents, who are not academics, that what a huge deal it is. But I know they're very proud, even though they probably don't quite understand what it means. But
well, if listeners want to read your article, it's on Nature, so it's open access. And the title of the piece is Genome of Middle Holocene Hunter-Gatherer from Wallacea. And it came out in August 2021. So before we even get into that, I think that you're definitely the first person on the show that I've ever had on to talk about DNA and genetics and ancient DNA. I've talked to a lot of people who are like functional morphologists or who study evolution, but like they look at like, I don't know, like the different structures, but I haven't actually talked to anyone who studies DNA. The closest I've gotten is some forensic scientists that I've had on. So I figured that maybe the first place we could start is talking a little bit about ancient DNA. And ancient DNA, well, I guess here's my first question. I actually got this question from my boyfriend the other day. I said, I'm going to interview this lady who studies ancient DNA. And he's like, is that different than modern DNA? So is ancient DNA different from modern DNA?
excellent question, actually, because of course, one of the most important parts of ancient DNA work is finding out if the DNA we're looking at is actually ancient or not, or if it's modern DNA. And so ancient DNA, as the word implies, just means that it's like genetic material that is from a person who's been dead for a while, for a long time, usually up to several thousand years, actually. And what makes ancient DNA different from modern DNA is that it's very fragmented. So the genetic material of you and me is like this very long array of bases, right? And like they combine and they code for everything in the body. And it's this very long molecule. But during the process of degradation over time, this long molecule is broken down into little pieces, which also makes it much more difficult to pick up with the technology that we have at the moment. And then there's also some chemical modifications that happen to the individual molecules that we can then use to differentiate ancient, so degraded DNA from modern DNA, which is most likely what we call contamination. So what's come onto the bone that we're studying after the person died.
Yeah, for sure. So the basic difference between modern DNA and ancient DNA is just that ancient DNA is, as it implies, ancient. It's just old. But like the things that make up the DNA are still the same. Like it's the same nucleotides, right? So we're gonna do a little very basic biology lesson, right? So DNA, deoxyribonucleic acid, is a double-stranded molecule, and it has base pairs that join up to get together. There's adenine and thymine. Those are always a pair, and then cytosine and guanine are always a pair. And those nucleotide pairs, it's basically just like billions of them, and that is what determines your genetic structure, which is crazy if you think about it. It's wild.
Yeah, just the different like order of what these nucleotides. So if it's cytosine, thymine, adenine, something, and then it repeats, and just the order basically defines what you look like, what organism you are, what everything.
Yeah, yeah,
Yeah,
yeah.
pretty crazy.
For sure. I mean, and so DNA is the reason that it's called a blueprint is because it's inside every single one of your cells, inside the nucleus, and it basically contains the instructions for how to create different kinds of proteins and other things that your body then uses and then builds you, which is wild. Um, and yeah, so different nucleotide groups code for different proteins, etc. Um, but yeah, so like the way that it works in ancient humans is the same that it works in us. It's just real old. And so one of the things you're talking about is degradation. So I think that anyone who's watched like a modern forensic TV show like Law and Order or whatever knows that we're like constantly shedding DNA. And you can pick up DNA from like a little bit of spit or like the end of a hair follicle. But if you're looking at something that was buried in a cave for 5,000 years, probably a little bit harder.
Yes, that's this thing. Like if you think about like like at the moment, like there's DNA everywhere, right? Like whatever you touch, and like you said, and for forensic science, that's really important that you leave DNA everywhere you go. But like as soon as the body dies, or as the DNA is not inside your body, it's not being repaired anymore, right? There's like repair mechanisms that constantly repair to make sure that your cells are working as they should. But once that stops, the DNA immediately starts degrading and like breaking up into smaller pieces. And then also in combination with UV light, UV is very, very bad for DNA. It just makes it fall apart into thousands of different tiny pieces. And also water, actually. So the DNA also reacts with water in a process called hydrolysis, which then also makes the bonds between the different nucleotides unstable and break apart. So once you're looking at a bone that's really old, then it's much more challenging to detect all the different small DNA fragments and then build them back together into this long molecule that is actually informative.
For sure. Yes. So yeah, the pro tip for listeners, anything that ends in lysis means that it's basically exploded. Um, so hydrolysis is just the bonds are breaking apart. And that's also why people tell you to wear sunscreen all the time, because UV is really bad for your DNA. So you have these like things that are actively degrading the DNA. And then on top of that, you have potential environmental contaminants.
Exactly. And that's the second biggest challenge that we have with ancient DNA is that, of course, if the DNA is so small, then any other DNA, so any person who touches or who comes in contact with the remains after is going to leave longer strands of DNA and much more that again then is easier to pick up than the tiny DNA fragments that we actually want. So yeah, avoiding contamination also from us as people who work in the lab with the fire is actually one of the most important things that we do in the lab. So we get really dressed up in like lab gear that looks like we're working with really intense chemicals, but we're not. It's just that we're trying to protect the bones from us rather than protect ourselves from anything on the outside.
So this is actually a really interesting thing. So you pull the DNA from the bone, but you're not like taking a surface sample because that's going to be the most contaminated, right? So where do you get the DNA from in the bone?
Exactly. So also, of course, usually the burials that we work with are in soil, and like also soil also contains lots of DNA, not just from humans but also from microbes and everything. So we also want to avoid contamination with microbial DNA. So when we sample a bone for DNA, we actually try to use one of the hardest parts of the skeleton, which is the petrous part of the temporal bone. So this is the bone, the part of the skull where your inner ear sits, so the cochlea is inside. And this is just a very dense bone also during your lifetime. And that's why we think that it's been shown that this is the part of the body where DNA preserves the best over time. And then again, to avoid the surface contamination, we tend to cut the bone open. So we sample in the middle, actually near the cochlea, so the inner ear, the spiral.
Mm. So we've talked to a lot of different evolutionary anthropologists. Well, the first person I interviewed for the show was Dr. Bernard Wood. And one of the things that he talks about a lot is that, especially with ancient bodies, you're not dealing with stuff like Homo erectus or anything, but with older bodies, you don't have as many body parts, right? Stuff doesn't preserve as well, which I can imagine would be a challenge for ancient DNA as well.
Yes, certainly. I mean, to find a complete skeleton in a burial is, I guess, very rare. So also finding skulls is, I guess, not super common. So the petrous part is definitely, at least at the moment, the preferred part, because that's where we so far have had the best success at finding ancient DNA. But also, generally, all bones contain DNA, right? They have to. So it's just that there's some parts of the skeleton which are more resistant, I guess, to degradation. And there was actually a study published, I think, last year by one of my colleagues who statistically investigated which part of the skeleton. Actually, they tested loads of different parts of the skeleton for DNA, and could actually show that the petrous part was still the most reliable one, but also teeth are quite valuable because, as we all know, they are also really hard, and they tend to survive because it's such a hard material. And yeah, there's also a couple more long bones that also in that study seem to be quite quite good.
Yeah.
It also always depends on the conditions on the site. Like, so far, we've not been able to build a model that can predict if there will be ancient DNA preserved or not. So generally, it seems that more temperate climates or even like permafrost are, as you might expect, quite good as preserving also DNA. And more tropical regions where also sometimes the soils are more acidic, and it's also more humid, then that usually makes it more challenging. So
For sure. For sure. And this is something that we've also heard from the forensic scientists who have been on. Um, so, okay, you found the bone. Someone found the bone, which is great. They've brought it to your lab. You've taken all of these measures to make sure that you don't cross-contaminate it. You've gotten your sample. Now what? How do you go about the process of looking at ancient DNA?
yeah, so actually, for to study the DNA, we actually only take a very small sample. It's only like around 30 milligram so of bone powder, so it's really not much. And then after chemical treatment, we isolate just the DNA from it. So all other chemical parts of the body are divided from the DNA molecules. And then this is also sequenced, which is quite a complicated process, but it basically translates all the nucleotides into letters that we can look at on a computer screen. So all the adenines and cytosines, then we can see them as A's and C's respectively on the computer screen if you were to look at the really long list of letters, basically.
For sure. And so when you do this, do you get a whole genome?
That does depend on preservation, but with ancient DNA, it's more challenging for sure to get the full genome. We usually work with SNP data, which means single nucleotide
Polymorphism.
polymorphism.
Yeah.
Yeah, it's a long word. But so we try to, because it's so difficult to get the full sequence, we try to focus and enrich specific parts of the genome, so this single sites, to make it more comparable also across with other genomes.
Right.
So rather than trying to fulfill the whole sequence, we try to find these around 1 million sites, which have been shown to be informative about the population history.
For sure, for sure. And just to back up and define what a SNP is, the single nucleotide polymorphism. So we talked about the nucleotide base pairs, like the adenine and thymine and the cytosine and guanine. And some people, instead of having an adenine at the top, it'll have like a thymine. So you know what I mean? Or you'll just have like a single like little variation somewhere. And a lot of the times it doesn't even really make a difference to the person, just as a living, breathing human being. But those little variations can show up in greater numbers in certain populations, which is how you can tell that someone might be from a certain population versus another population. So you're just looking for those little variations, and it's so small. It's a single nucleotide polymorphism, a SNP. It's wild.
Yeah, for sure. And like, of course, like the majority of the genome we all share as humans, right?
Hmm.
It's really a very, very small percentage that is different between people. And what's also interesting about that is that actually the variation within a population is actually usually larger than the variation between different populations. So really the genetic differences between different people are really much smaller than some people might think.
Yeah, I think definitely a good thing to keep in mind in today's world. So I guess we've started to kind of get at it, but why study ancient DNA at all to begin with? Especially if like people are probably like, wow, you can't even see the whole genome. So what's the point?
Yes, that's a very good question. But I think that ancient DNA is another puzzle piece, I guess, that helps us understand what what was going on in our past. And there is, of course, archaeology and also anthropology who've been teaching us a lot about what people in the past were doing, what they might have looked like, and what kind of social relationships they might have had. But I think with genetics, it's just a slightly different viewpoint, trying to get at the same kind of questions. And I think it's very valuable, especially also about periods of time where we, for example, don't have any historical documents to tell us what might be going on. And yeah, it's kind of, I guess, yeah, it's just a different perspective. And when we bring all of these perspectives together, so mainly, I guess, archaeology, genetics, and also linguistics, historical linguistics, then in my opinion, that brings us a more, I guess, complete picture about the past.
For sure. For sure. I think one of the greatest contributions that ancient DNA is able to give us is an idea of migration patterns. Understanding how humans have moved through the world and how different populations are related to each other. So we get a better idea of what that, I don't want to say evolutionary past because it's not that. It's like what our ancient, but still modern, anatomically modern human ancestors, what their past would have looked like. And I think one of the other things that people are really interested in, and you see this with like 23andMe and stuff like that, is us crossing with other species that are no longer around, or so like Neanderthals or Denisovans, which are genetically distinct, but we carry some of their DNA.
Yes, and especially for the Denisovans, we wouldn't actually know that they existed really without ancient DNA, because the only archaeological finds we have that we now call Denisovans are finger bones, pretty much. So very small bones. I'm sure all the anthropologists in the audience know that they are very small, and they've only been identified at two different sites, one in Siberia and one in Tibet. So the Denisovans are actually, you couldn't tell that they were different species because you only had the very small bones, which are not, I guess, very informative
Hmm.
morphologically, at least that's my understanding. But when this was sequenced, when the genome was sequenced, this was already, I guess, in the mid 2000s, we could actually show that the bones there were not modern humans, like one would have expected, or even Neanderthals, which was the only other archaic genome that we had at the time. But that these bones belonged to a group of hominids who were related to Neanderthals more than to anatomically modern humans, but still distinct from them as well. We could then also trace this ancestry into people who are around today, specifically also in near Oceania. So back in the day, I guess these Denisovans must have had a much larger area of occupation than we can find now from archaeological finds. But they definitely left fingerprint in present day genetics as well.
Yeah, genetic fingerprint for sure. So this is kind of like how people in Europe might have a higher percentage of Neanderthal DNA than people who are originally from, say, China. Likewise, people who are so Australian Aboriginals, um, and I believe there's like a Filipino population and another, I can't remember. They have a higher percentage of Denisovan DNA than other people do. And when we're talking about a higher percentage, we're talking about like 5%.
Yes. So actually, all people outside of Africa have a similar level of Neanderthal DNA, and that which I think like the average is around, I think, 2.5%. So also really very small. And then the Denisovan ancestry that we can find today in people from near Oceania is around like 3 to 4%. So
Yeah,
it's also
it's
very small, but certainly, yeah, just poses some really interesting questions about what like the settlement history also of the world, and at what point did our ancestors interact with Neanderthals and Denisovans, and in what context. Yeah, it's just, yeah, fascinating, really.
a
Like
lot of
humans
exciting
never just
questions.
existed. Yeah, they never just existed like by themselves and never met anyone, right? They were other hominids around, and clearly like they had children together, like we know that. So it also, yeah, poses questions about like how these different hominids interacted, and yeah, it's just very, very fascinating still.
I know, you start thinking about it and you're like, I don't know, maybe there's a market for like some Neanderthal human romance novels. I don't know.
Maybe.
Okay, so I think we've done a really good job of discussing a lot of different background stuff so that we can discuss your paper.
Yes, because actually also in the individual that we studied, which is around 7,200 years, and was found on Sulawesi, which is an island which today belongs to Indonesia, we actually also found Denisovan ancestry, which was very, very exciting.
Why was it so exciting?
Yeah, because of the limited range of where we find Denisovan ancestry today, and also because, yeah, there's this question also of how did modern humans earlier than 50,000 years ago come to Australia and to what we know today as Papua New Guinea? Like, how did they did they cross from the mainland to the islands? Because Wallacea, which what's kind of the, I guess, biogeographical name of this region, this is a region between the Southeast Asian mainland, so Thailand and Vietnam and Cambodia, and Australia and Papua New Guinea, where these islands, such as Sulawesi, they've never been connected to the mainland, even when the sea level was much lower during the last ice age. So they've always been separated, which means that when our ancestors crossed over towards Australia, where we have evidence dating back to 50,000 years ago, that means that they must have crossed over the open ocean. And yeah, that's just quite fascinating to think about. And I think it also implies something about their level of organization and just technology also to be able to make that crossing. And there's still different hypotheses if people back over 50,000 years ago, if they came across Sulawesi to Papua New Guinea and Australia, which at the time were a big supercontinent, or if they came along the southern Indonesian island chain, which includes Bali and Timor and other of these small islands there. So to find Denisovan ancestry in this individual, it just also shows that, there must have been interactions with Denisovans either in Wallacea or on the mainland previously, but certainly it shows again that the range of the Denisovans was much larger than you would think. And if they also existed in Wallacea, then they must have also crossed the ocean to get there. So just
That's awesome.
again opens up new questions.
No, that's really awesome. That I'm so excited to see what more research comes out of your PhD, because this is such an exciting first thing. I want to kind of switch focus a little bit to something it's a hot topic in all of anthropology, but genetics has its own, archaeogenetics has its own set of concerns, which is the ethics of archaeogenetics. I think maybe, well, let me ask you, what would you say some of the concerns are in archaeogenetics?
I think what's most critical with any genetics work, and especially also with ancient DNA, is that the genetics are kind of like a really essential part of a person. And when we then go from genetics and try to talk about someone's ancestry, and we can also talk about like genetic kinship, so who they were related to and stuff, it's very connected to human identity. And of course, since these are usually archaeological, well, always archaeological finds, like we can't go and ask the people for consent, right? Like you could for genetic studies, which you do today. You have an ethics board and you have to get informed consent of all of your participants. And it's a really controlled process, and that's really important also. But with ancient DNA, it's more challenging because, of course, the people have been dead for thousands of years. So there's also responsibility of us scientists to go out and find and consult with people who today feel a connection with these as their ancestors. And because these like people in the past, so our ancestors, they don't just exist, like they existed in the past, but in a way they still spiritually exist today and are relevant. So there's like a responsibility not just for the deceased, but also to people nowadays who are still alive who have a connection with these individuals.
For sure. This can be a really complicated conversation. I think one of the most famous examples in the US is Kennewick Man, which is a skeleton that they found in Kennewick, Washington. We're not super creative when it comes to naming things. And the sticking point of this skeleton is that it was around 10,000 years old. And there are a lot of active local Native American tribes in that area who claimed a kinship to Kennewick Man, and they wanted the bones repatriated and they wanted to bury them. And then on the other side of this equation, you had scientists who were saying, well, Kennewick Man, probably not related to y'all. Like, I know he was found where you currently reside and where you've historically resided for hundreds of years, but he probably has no relation to you. He's more closely related to some other group. So, like, really, we should be asking some other group. So it quickly becomes a very complicated situation. Um, Kennewick Man was discovered in, I want to say 1996, and which was around the time NAGPRO was passed. So we're still seeing like a lot of that conversation kind of coming out. And I believe most of Kennewick Man is still in a museum vault somewhere because they're still kind of figuring out what they want to do with it. No, it does become very, very complicated. And a lot of it comes down to something that we talked about a little bit before the show, which is community engagement. Like, how do scientists talk with communities? How do they form a rapport? Is there a way for both sets of people to be happy? Are those things mutually exclusive, their end goals?
Yes, exactly. And I think what you said about is exactly the point. And like, just because, I mean, if we go only a few generations back into the past, then suddenly you have so many ancestors that you share with so many other people that, like, direct genetic relationship might not be there anymore. But I think that it's really important to take into account that as humans, we don't just exist as our biology, right? We are that what makes us is actually not our biology, right? It's what we think of ourselves as our identity, which group we might feel connection with, what we identify as. So I think that's precisely why engaging with people who feel a connection and who identify, who have these ancestors as part of their identity, also as a group identity, is really, really important. And I think there's nowadays also there's also an emphasis on bringing indigenous people into genetics so they can be their own spokespersons, because it is a perspective that's difficult to understand. I mean, especially like speaking as a European, like that's, I just think that we have, as scientists generally, we have this responsibility also to consult with people like before studies are done, but then also to bring back the results to the community, share what we've actually found. And this means going out and probably breaking down the research into more understandable terms. But I think it's a really important part, and I do wish that there was some more emphasis also on that part, and some more, I guess, training also to scientists to do that.
For sure.
So far, I think there's, especially also in ancient DNA, there's been a lot of criticism from bioethicists, but also archaeologists and anthropologists, that archaeogenetics is going too fast. That that archaeogenetics as a field is just going and sampling all the bones to produce as much data as possible. And I do think that it's valid criticisms. I mean, I'm a PhD student. I started 3 years ago, but I think that as a field, there is definitely a responsibility that what we do is sustainable and also ethical. And I think there's still room for improvement.
Yeah, no, definitely. And this is something that anthropology has been grappling with for a super long time, and we still are. And there's still a lot of conversation around it. And there are no easy answers, I don't think, unfortunately. But it is something that's been a theme throughout this entire show. We talk about, because most of the people that I interview are in the United States, we talk about the Native American Graves Protection and Repatriation Act, which I earlier referred to as NAGPRA without defining. We talk about that all the time because it's like it's just super relevant to what we're doing.
Yeah, and I think it's also, but we also have to remember that not many countries have something as NAGPRA, right?
Right.
Like it's quite a unique piece of legislation, but that of course doesn't mean that the principles of don't count in other countries. Just because some would be legal there doesn't mean it's ethical, right? So
for sure,
I think there's now also a lot more legislation also coming in. So there's protection for artifacts and human remains, which I think is really important, that the countries of origin have a say in what happens. Otherwise, it'll turn into like kind of neocolonialism, since most of the big ancient DNA labs nowadays are again in Europe and the US. So
yeah. I mean, we're also dealing with this with bones. At the Smithsonian, they had quite a large collection of skeletons from overseas as well. And it's like, once NAGPRA got passed, people started thinking like, well, what about all these other skeletons that we have here? I mean, yeah, like we're not like by law, according to NAGPRA, required to do anything with them, but like maybe we should be thinking about returning them.
precisely, there's so many anthropological collections that also date back into colonial times, where even if at the time they may have been acquired through at the time legal means, that most certainly is a red flag nowadays, because I mean there was all these power dynamics at work that even if there are, in my opinion at least, if there's records of financial transactions, that doesn't mean necessarily that this was a voluntary action. So all these big anthropological collections also in Europe, like I think some people consider it like a really valuable resource also to like learn more about the past. But I think also the context of how these bones have been acquired is really, really important. I think I wouldn't want to work with bones from such collections if it's not towards repatriation. But yeah, so there's still also a lot of work to do there too.
So I guess the question is, how does this relate to the study that you did most recently?
So in this study, actually, it was a collaboration between archaeologists from Indonesia and Australia, and then we came in to do the DNA work. But also the archaeologists in Indonesia, I mean, it was also there very important to talk with people who actually live in the area now, for them to understand. And actually, they actually gave nickname to the individual that we studied, Besse, which I think is an endearment name that you use for young girls.
Mm-hmm.
While generally I think like assigning people names after they've died just feels strange, but in this case I think it shows that they still feel like it's part, like this individual is part of their community. So I think that was very valuable to have people from there involved in the study and also in the analysis and interpretation.
Yeah. That's all around a really interesting, exciting piece of work that you've done. And I'm super impressed. I'm sure everyone that you work with is also super impressed with you. I want to be respectful of your time, talking about respect. So I'm going to wrap up the interview with the final 3 questions that I ask every guest. So the first question is, if someone wanted to learn more about the work you do, what would be a good thing for them to read?
Of course, the first address would be the paper that came out
Hmm.
in August. Like you said, it's open access in Nature, so it's available for everyone. And then we also actually just submitted a new paper today, so that's very exciting.
Congratulations.
So I guess that's the next things. They're like, stay tuned, I guess, for more from that region of the world. And I think there's also generally, recently there's been also some more discussions about the ethics of archaeogenetics, also published in bigger journals. So there was a paper actually that came out last week, which also in Nature, about the ethics of DNA research on human remains. But there was also a lot of criticism associated with that paper, including from my side. So there's also last year there was a paper by Wagner et al. More on the North American context, if people are interested in that. And
I think that one of the things that's really important to do in science is, especially when you're getting kind of like these opinion pieces, is to read the pushback on it and figure out where you fall on the spectrum, right? Because there's very rarely does someone put out a paper and everyone's like, "Yep, sounds good. No problems with it." It almost never happens. So understanding like what the responses are can give you a better idea of like what that conversation actually is. And I'm also going to put in a plug for there's an open access book that was created by the American Anthropological Association. It's called Explorations, an open invitation to biological anthropology. It's completely open source. And there's some really, really good chapters that explain DNA, evolution, migration in there. So, like, if you just need like a basic intro to all of this, I think it's a great place to start. And it's completely free, it's completely online. You can download it. There's teaching resources, there's a lab. It's pretty cool. I was really excited to find that.
one of my colleagues and myself actually also gave an introductory lecture on archaeogenetics as part of the summer school we organized
Mhm.
over summer, and it's available on YouTube as well. So I can give you the link
Yes.
if you want
If
to,
you send
if
it to me,
anyone's interested.
if you send it to me, I'll put it up on the website.
Of course. Like
Uh,
there, we also go into like DNA and the structure and what we can learn from archaeogenetics. So
yeah, awesome. Awesome. That's perfect. Okay, second question. What are you reading for work right now?
for work at the moment, it is this ethics piece and the responses to that recently, and then also I'm working on a new project. So I'm looking into the prehistory of Thailand actually, and I think there was also a paper recently that came out about some really old cave paintings in Wallacea. So pushing back, I guess, the human occupation there even further. So that's also very interesting.
Ah, so interesting. And then last question, what are you consuming for fun right now? Like it could be a TV show or a book or whatever. Someone said hiking the other day.
Oh, very nice. So I've actually gotten back into also reading for fun during the pandemic. Funnily enough, I had time to read again, which has been very nice. And I recently read the Broken Earth trilogy by N.K. Jemisin, which I devoured the 3 books in, I think, like 5 days. But I would really highly recommend them. They're kind of, I guess, so if you're into fantasy, but it's not high fantasy, it's kind of a, I guess, new fantasy in a way. It's not quite so tropey, and it's really a breath of fresh air, I guess.
I've heard the books are incredible. That's awesome. Okay, well, thank you so much for sharing your time with us. If you ever want to come back and tell us about the exciting things you're doing, you're more than welcome to.
Yeah, I'd be really happy to. I'm also happy for people to send me questions on Twitter or emails, and I'd be happy to answer everything.
Awesome. Well, have a wonderful day. It was so lovely talking to you.
Thank you so much.
Thanks for joining me for this episode of the Anthrobiology Podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show is produced by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.